Okay, so chapter 11, we will discuss this technique in sequencing, right? During this pandemic, like you see the importance of a DNA sequencing. Without this NGS, it would not be done, and then we will not be able to develop this messenger RNA vaccine at all, okay? So that's why this is so important now. So now, the DNA sequencing. From the very beginning, okay, the early days, the first technology that developed is called a chain termination sequencing. Basically, you use the single-stranded DNA molecule as the template in front layer, and then you perform the DNA synthesis, and then run the polyacromide gel so that you can determine the sequence. Okay, that's chain termination sequence. And another method is called pyro sequence. All right, so that's the later on that people develop pyro sequencing. Okay, and this is the kind of a result, a typical result that you in old days that people can see for the chain termination sequencing, and then you try to decode the DNA sequence. Okay, the basic principle for the chain termination sequence is here. So as you remember, the deoxynucleotide, all right, we have 3' hydroxy group here, okay? But for the chain termination sequencing, you have to use a dideoxy, okay, dideoxynucleotide. So dideoxy, that means the 3' hydroxy group has been replaced by the hydrogen ion, all right? So this oxygen has been removed, and so it's only hydrogen here. So the impact would be if you use a dideoxynucleotide. That means during the DNA synthesis, if you incorporate this dideoxynucleotide, then the synthesis will stop. Okay, will no longer to continue, right? So on the right side, this is just a review from the DNA synthesis. Okay, remember, so these incoming nucleotide, right? When they come, so these oxygen here, the hydroxy group, will perform the SN2 reaction, attack this alpha phosphate, and then remove this pyrophosphate, right? Pyrophosphate, and then link this phosphate to this oxygen, okay? Now, if you replace this oxygen with this hydrogen, now that's dideoxy. Therefore, there's no oxygen available to perform the SN2 reaction. Therefore, this incoming nucleotide will not be able to bind, all right, will not be able to form the phosphodiester bond. And then the synthesis will be terminated. So that's why it's called chain termination sequencing. And during this process, another thing is important that you see the pyrophosphate, this pyrophosphate is released. This is important, all right, for another technique. It's called pyro sequencing, all right? The pyro sequencing basically use this phosphate to perform the reaction, to give it a signal, to tell you which nucleotide has been added. So this is also important that you need to know. During this whole process, the fundamental theory is the same, all right? The DNA synthesis, you need to have this oxygen, all right, the hydroxy group to form the phosphodiester bond. If no hydroxy group, then no DNA synthesis. And during this process, also they release the pyrophosphate. And this pyrophosphate is important for another technique, okay? So this is the DNA synthesis process. And for the detail here, so basically, dideoxynucleotide can be the ATP, CTP, GTP, TTP, dideoxy, right? But for each reaction, for the chain termination method, you need to perform 4 different reactions. Each reaction, you will only add one type of dideoxy NTP, all right? So you can have DD-ATP in one reaction, DD-CTP in another reaction, DD-GTP in third reaction, DD-TTP in fourth reaction. And in these 4 different reactions, they will perform this DNA synthesis. And then during the synthesis, they will incorporate they mix with the regular deoxynucleotide. All right, and then accidentally they might incorporate the specific dideoxynucleotide. Once they incorporate the dideoxynucleotide during the DNA synthesis, then the DNA synthesis stop, is terminated, so that you have the length of this fragment, right? So for example, in this case, you use a dideoxy ATP, and then there are 3T. So for this fragment, basically here they incorporate the regular DA, and then here DDA, and here DDA, and then DDA here. Then you have 3 different lengths of DDA. Then you perform the gel electrophoresis based on the size of the fragment, so that you know the specific location of the A, all right? Specific location of the A. And again, for the second reaction, if you use this DDCTP, then you will know specific location of C, and DDGTP, specific location of a G, DDTTP, specific location of T. And based on this experiment, so that you run these 4 different reactions, and then each reaction you run in one lane of the polyacromide gel, and then you see different fragment from the size of fragment, and then you know the location of the specific nucleotide. So for example, in this lane, you use a dideoxy ATP. So the band here will tell you the location of A. And here with dideoxy CTP, then the location of C. And then you can read the sequence. So from here, from the bottom to the top, that'll be a CGATT. All right, that's how they read the sequence. All right, so that's in the early days, like 20, 30, 40 years ago. And later on, then people develop another technique. All right, when the scientists developed this capillary electrophoresis (CE), and then they incorporate this dideoxy nucleotide with different fluorescence color in here. So let's say dideoxy G with black color, dideoxy A green color, dideoxy T with the red color and pink color. So then they perform the reaction together. All right, no need to perform the individual reaction like the long time ago. And then they perform this reaction together. Then they will have different lengths of the fragment with different color, and each color will represent the location of that specific nucleotide. And after the reaction, they perform the capillary electrophoresis. So the capillary electrophoresis, they will arrange the fragment from the smallest size to the largest size and go through the detector, so they can detect the color. So from the color, then you will know the sequence. So black is G, and then the next one is A, and then C, and G, and C. So that's the current technology. Use the fluorescence to determine the DNA sequence, but still use the Sanger sequencing, which is a chain termination sequencing principle to perform the DNA sequence. Okay, so this is one method. And then another method that's called the pyrosequencing. Yes. No, it's not. It's not a chain termination sequencing. It's called Sanger. It's Sanger sequence. Sanger, Sanger sequencing. All right. It's not chain sequencing. It's called Sanger sequencing. Okay. I wish. All right. So it's a Sanger sequencing, and then later on, then scientists developed this pyrosequencing. All right. Pyrosequencing basically based on the principle that I told you earlier, that during the DNA synthesis, pyrophosphate will be released. All right, pyrophosphate will be released. Basically, the pyrophosphate released, then they cannot be that can be converted by the enzyme sufferers into a flash of a chemiluminescence, and so that you can detect a signal to determine which nucleotide is added. So in the early days, very early pyrosequencing, you have to perform this one at a time. So they say added the DATP first with the orange color, and then it didn't work, all right, can be degraded if it did not, since it did not add it to the nucleotide, did not add it to the template. So it degrade, and T now work, degrade, and G incorporated, and then release the pyrophosphate, and then the pyrophosphate will give you the signal. Then you know G is added, and then you continue the cycle, all right? So that would be this principle here that you see the take the alpha phosphate, release the pyrophosphate, and then this pyrophosphate, okay, pyrophosphate here in the presence of the ATP suffrase, then they will combine with the adenosine 5-phosphate into the ATP. Then this energy then will push the luciferin, okay, to convert to the oxyluciferin in the presence of luciferase, and then give you the signal. All right. So because these pyrophosphate and then ATP suffrase and turn to the ATP, and this energy provide the luciferase to turn this luciferin into the oxyluciferin, give you the light, and so that you know a specific nucleotide is added. So that's the principle of the pyrophosphate. So those are two fundamental basic technology that in the early days that people use. That's also called a first generation sequencing. And then later on, because as you may know that in the end of the 20th century, in 1990s, that there's a project, Human Genome Project. They try to use this technology to sequence the human genome. And then like, it takes like 10, 15 years until 2003 to finally make the human genome. But during this process, the scientists continue to develop the sequence technology. And then in the early 2000, in the between 2000 and 2010, then there are 4 different NGS technologies have been developed. One is called massively parallel pyrosequencing. Okay, and the vendor will be Roche. And then sequencing by synthesis, by Illumina. Sequencing by ligation, that's Applied Biosystem. Sequencing by the semiconductor technology. Okay, so these 4 different technologies has been developed. Okay, so of course the pyrosequencing, the idea would be from the pyrosequence. Okay, so for the pyrosequencing, so you have the beads are attached to fragment the genomic DNA, and then you use a PCR to amplify first, and then in the separate water droplet, and each one will load into the multi-well here, into the well, and then from the well, each well they will perform the pyrosequencing. So that's why it's called parallel pyrosequencing and massively, because you can have the hundreds, thousands well to run the sequence at the same time. Okay, so that's a massively parallel pyrosequencing. And then we can watch this video quickly so you can get some idea.
Hey everyone, quick biochemistry basics here. Let's talk about pyrosequencing. Pyrosequencing is a sequencing technique in which incorporation of DNTPs in the DNA is detected in the form of light. Let's see the concept of pyrosequencing. When DNTP is added on the template DNA, pyrophosphate is released. This pyrophosphate can be converted into ATP by the enzyme ATP sulfurylase. The ATP can be further utilized by luciferase enzyme to generate light. This is the same enzyme that generates light in fireflies. Along with ATP as an energy source, the luciferase also uses oxygen and luciferin as a substrate. Now here comes an interesting question. Addition of any DNTPs, A, T, G, or C, will generate pyrophosphate. So the question is, how do we detect which nucleotide is added? Well, to solve this problem, the reaction is carried out in a flow cell where only one DNTP is added at a time. The flow cell is controlled by the computer, which knows which DNTP is added. Now, because the reagents are coming in and out of the flow cell, the template DNA can be easily flushed out of the cell. To avoid this, the template DNA is immobilized on a bead which remains in the flow cell. Also, there are many copies of the same template DNA generated by the PCR. Because of this, when dNTP is added, large number of pyrophosphate is released. Hence, the light produced will be intense and easily detected by the sensors. If dNTP is not incorporated, no light is detected. Hence, the sequence information of the DNA can be easily obtained by detecting the light. It should be noted that there can be unused dNTP in the flow cell. This unused dNTP is removed by apyrase enzyme. The more advanced pyrosequencing also allows sequencing of different templates in the single run. For this purpose, the beads having different template DNA are kept in a picotiter plate. All the template DNA are PCR amplified and immobilized on a single bead.
So basically, the fragment of DNA will be amplified by PCR first and then attached to the beads, right? Attached to the bead and then PCR amplify, and then each well will have one bead. That means each well will have different sequence, all right? Different template. So that's why you can perform the massively parallel pyrosequencing.
Each well of the picotiter plate will have a single bead with many copies of the template DNA. The picotiter plate is then kept in a flow cell while the reagents are added and flushed.
Okay, so that's the principle for a pyrosequencing. And then the next technique is called a sequencing by synthesis. And this is a reversible chain termination. Okay, this is like a modification technique from the chain termination technology. So basically, each nucleotide, of course, they carry with different color of fluorescence. And they are the reversible chain terminator because they at the 3' end, they have the blocking group. And in the nitrogenous space, they have this fluorescence dye. And there's a cleavage side. So every time when you add a nucleotide, if this nucleotide is attached to the template, then the 3' blocking group will block the next nucleotide to come. All right, but in the meantime, you will detect the signal from the fluorescence through the color, and then after you detect the color, then you will cleave these fluorescence dye and the blocking dye. Once you cleave these two, then this oxygen will be available to attack the incoming nucleotide. All right, so that you can continue to repeat this process to add a nucleotide. So that's why it's called sequencing by synthesis, and also it's you use this reversible chain terminator. Okay, and this will be a little bit more complicated for this because this is a very popular technique nowadays that in the industry. Okay, so I'll let you watch this carefully.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands.
So adapter region. So basically in between here, that's your template DNA that you want to sequence. Then you attach the adapter, okay? Adapter.
A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments.
So this step is just like in the parallel sequencing, that the first step you try to PCR amplify the sequence, okay? And so they use this clonal amplification so that you can duplicate many templates, and so that you will not lose the signal, okay?
After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read.
Here's a 3' end, all right? So you bind to the 3' and then you do the 5' to 3' synthesis.
With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome.
Then this part would be like do a sequence the genome, because from all this read, then you align to this reference genome, so they can, and you can generate this genomic sequencing.
For variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared.
Okay, that's a sequence by synthesis. And by ligation, it's more complicated, and this is the only sequencing technology that you don't need to use polymerase because you only need a ligase, all right? Only ligase is required for sequencing by ligation. For this sequencing technology, you need to have a set of 16 different oligonucleotides because that can represent, like here, 16 different oligonucleotide that represent 16 different possible combination of the two dinucleotide. So from AA, AG, AC, AT, all the way to TA, TG, TC, TT. Okay, and then for these, basically, you have these two nucleotides that's for you to determine, and then you have 3 degenerate nucleotide and then a cleavable nucleotide. So basically, you have the adapter sequence and then you have template, all right? And then during the reaction, you mix the template with these 16 different set of the oligonucleotide. And only one, they say here is CC, therefore only GG will bind here, all right? So GG bind, all right? And then with this signal, you can record it. Once you record it, then you cleave this 3Z here. And then you add another set of 16 oligonucleotide, and then CA here, only the GT will bind, all right? So by doing this, then you can determine 2 nucleotide and then 3 gap and 2 nucleotide and 3 nucleotide gap. And that's the one cycle to determine every 2 nucleotide in 5 nucleotide. And then when you finish the first probe, then the first primer is done, all right? Then you move on to the second primer. For the second primer is designed is 1 nucleotide shift, right, to the left. So as you can see, for primer 1, it was ATC, and then primer 2 is on AT, so that you can determine this nucleotide now. So again, you perform the same ligation experiment. So then you can slowly determine these sequence. So for this ligation sequencing, you need 5 different primer in order to cover all the sequence, okay? And the sequence by the semiconductor. So basically, this is the reason why, because when you perform the DNA synthesis, every time when you incorporate the nucleotide, not only you release the pyrophosphate, but also you release 1 hydrogen ion, okay? Remember that the hydroxy group, 3' hydroxy group, when you, the oxygen attack the phosphate to form the phosphodiester bond, the hydrogen from the hydroxy group will be released. Therefore, if you use this semiconductor technology, then you can detect, okay, the presence of the hydrogen. And then the presence of a hydrogen, then they will change the pH so that they know which nucleotide is added, okay? That's by the semiconductor technology. Okay, so I give you this overview of these 4 different technology.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is the gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from the 3' end.
deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1, they lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2, each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab joes or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera, which records the appropriate sequences of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods, since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end, that are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the two probe-specific bases and upon annealing is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last three bases, the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by one base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing rates generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution, and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques. In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome and enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina high-seq machines. The ion proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next.
So that's basically the next generation sequencing. Okay, any question?